And what I'm going to be talking to you today about is some work that I've been doing with some collaborators on detecting hybridization using phylogenetic invariants. So phylogenetic invariants were first introduced in the 80s by a pair of papers by Kaplan and Felsenstein, and then another by Lake. And an invariant is basically any function that takes a tree topology and then the model parameters that are on that tree and returns 0. And sometimes there are particular invariants or particular functions that will be 0 only for a particular tree topology and not for others. And so we call those phylogenetic invariants because they have phylogenetic information about the species that we're looking at. And basically the way that we actually construct these when we think about looking at our own empirical data is we use the distribution of site pattern probabilities at the tips of our trees. So when they first came out, they were thought to be not all that useful because there are a lot of different site pattern probabilities that you can actually observe, and if you don't have enough data to estimate them well, then they're not particularly useful. I think it's fair to say that that's not really that much of a problem anymore because we have full genome data sets for a lot of taxa, so they're hopefully becoming much more common to see. And the nice thing also is that estimating this distribution of site pattern probabilities is really fast, even for huge data sets, and then using them to make these phylogenetic inferences can be done very quickly by just comparing these frequencies. Another thing to consider, though, is that we know that different regions of the genome have different histories that might be different from the overall underlying species history. And so we think typically about gene trees evolving within species trees. And so here we have the lineage sampled in species 1 and species 2 coalescing for the lineage from species 3, which doesn't match the species tree. But then on top of that, we can have mutations that occur where species 1 and species 2 have the same nucleotide because of a single mutation, rather than what you might expect with species 2 and species 3 being sister to one another. So the way that we can develop invariants that actually sort of tell us about the species tree topology rather than just a single gene tree topology is to consider all the different topologies that a gene tree could have, plus the site patterns that would arise on them, and then we can get an estimate of what the species tree might be. And if you're familiar with the software SVD quartets, that's basically exactly what this program does. It will give you the unrooted species tree topology for a quartet of taxa. So adding hybridization on top of the coalescent is basically just to take a network like we have on the left here and consider that it's basically a linear combination of two possible parental species trees, where the hybrid either goes with parent 1 or with parent 2, and the probability of a lineage being sampled and then going with parent 2 is gamma, and then the probability of that lineage going with parent 1 is 1 minus gamma. So invariants that you may have heard of, so I would imagine a lot of people are familiar with these ABA-BABA tests for calculating the D statistic. This is actually using an invariance-based test, where the probability of seeing an ABBA pattern versus a BABA pattern is expected to be equal under this type of model when there's no hybridization. Another more recently developed test is called the Hiltz test, which was developed by Laura and Julia, who I worked with for the stock, uses a similar sort of style of invariance, so you can see the numerator of these two are the same, but the denominator in their test is different, but the sort of alluring thing about it is that it's directly a function of the hybridization parameter that we're going to be interested in estimating in. The nice thing also is that both of these use the normal distribution, so we can do nice statistical tests. But one of the big things that these sort of models assume is that once hybridization has happened, it's sort of a uniform process within the hybridized population. So basically all of the individuals within there should be equally introgressed, which is not typically what we see in nature, at least for episodic gene flow or things like that, where only some individuals might have the introgressed haplotypes and others might not. So what we wanted to do was sort of test how robust are these methods to actually detecting that, this is not actually a hybrid species, or the process is not uniform versus just one individual being introgressed. So we did some simulations where we simulated sequence data using MS and SeqGen under the GTR plus I plus gamma model. And we did a 4-taxon species tree, so an outgroup with 3 in-group taxa, and we varied the depth of the tree in coalescent units. So basically we had a short tree, so a lot of incomplete lineage sorting versus a long tree, and then one in the middle. And we varied the number of sites from 50,000, so that's 50,000 independent gene trees simulated on the species tree, all the way up to 1 million. And then we used SeqGen and scaled everything by 0.1 mutations per site, and we did 5 individuals within the in-group, and then 1 individual for the out. But the thing that we did to sort of demonstrate what might be considered an extreme case of just introgression was only 1 individual in the hybrid, in the species that we're going to be testing as a hybrid, was actually a hybrid. So we simulated it as an F1, and everything else had a gamma of 0. So it was totally related to parent 1, and no relation to parent 2 at all. Okay. So just to set up these results, on the y-axis we have the test statistic, on the x-axis we have the number of sites, and this red line is the critical value for the normal distribution for the test that we're going to be doing. And sort of our naive expectation would be, I think, that as you get more and more data, you sort of do a better job of saying, hey, this is not a hybrid species like we're sort of testing right now. So things would go down and stop being significant. What ends up happening, actually, is that as you get more data, you're more and more sure, and your test statistic gets bigger and bigger and bigger, that these things are hybrid species, basically. And so the test statistic will basically always increase as you get more and more data. And I think it's fair to say that that is not good. And so in this case, for this particular hypothesis test, we're rejecting the null hypothesis, but we're misinterpreting the underlying pattern, which is that there's only 1 hybrid, and the entire population is not hybrid. So what can we do? We messed with a couple different things. One of the most obvious ones is, well, you can just run a test like this, Avobaba or the Hills test on each individual, and that seems to work pretty well. For these tests, you tend to have a lot of data for them to work well, so we sort of wanted to preserve the amount of data that was originally in the data set that you were using to test it. So one of the things that we did was thought about some type of resampling approach, where you resample individuals within taxa. And when you do that, for this example that I'm going through, there's this really cool pattern that happens. And we call it hybrid wobble. And so what happens was we did 1,000 bootstraps, and the pattern that you're seeing is really indicative of the fact that there's a peak right around 0, and so that's all the bootstrap replicates where the hybrid was not sampled. And so everything that you're seeing is not significant because none of the individuals are hybrids. The next peak is when the hybrid was sampled once. The peak after that was when it was resampled twice, and if you did enough bootstrap values, you would get it for all the way from 0 up to 5 times. And so it's a really nice visual pattern saying, hey, this is not a uniform process that's going on in the population that we're looking at. So to sort of test these in some empirical examples, we wanted to see if we could find a data set that had multiple individuals within a couple of taxa. And so one of the ones that we pulled out was this Heliconius butterfly data set from Martin et al., just a paper in genome research. And they had whole genome data for 3 species. So for the in-groups, it was 4 individuals each, and then just a single out-group taxon. And the data set was huge, through 250 million sites. And we ran our hybridization detection analysis on the in-group and considered all possible different combinations of the 3 in-group taxa. I did bootstrapping as well, and I only did 20 reps. And as you can see here, the whole analysis took about 6.5 hours, which I don't know, for 250 million sites, I feel like that's pretty good. So the reason I only did 20 was because I honestly thought it was going to take a lot longer than it did. But what you see for the distribution of the test statistic, first of all, you probably can't see these numbers, but they're ranging between about 450 to about 500. And so the null distribution is centered at 0, and these are at 500, so you get a highly significant result. And because there are so few bootstrap replicates, I feel like this is not as useful as looking at the actual distribution of the gamma statistic that you're estimating for these particular hypotheses. And so gamma can only go between 0 and 1. And so if there are sort of hybrids included in some and hybrids not included in other, you would expect a bimodal distribution of some sort. But here we're seeing that everything is sort of nice and uniform. And so this looks like it's actually a case of what would be considered more like hybrid speciation rather than some type of just introgression that's not uniform in the hybridizing population. Another example that we wanted to go through was what if you had sort of more phylogenetic level sampling, so you only have one individual, but you have like maybe a species tree estimate for a lot of different species. So we got transcriptome data from these 26 species of swordtail fishes, and this is from a paper by Cui et al. From Evolution in 2013. So there were 26 species in a single outgroup, and there were about 3 million sites for this. And we ran all possible hybridization detection analyses on this, so all triples in all directions. And then to sort of control for false discovery rates, we corrected the p-value so that it was 9.6 times 10 minus 6. And the analysis took a few hours, so it wasn't too long or anything like that. One of the things that I noticed when I was looking at this is that there's a lot of variation in the value of the test statistics. So it can range. These are only significant values, but it ranges from something that's just on the edge to all the way up to 120. So there's a lot of variation, and so you kind of have to be diligent in your interpretation of how these things are going. The other thing that I wanted to point out is when you look at significant results for the value of gamma, we are picking up that there is a lot of hybridization in this group, which is what would be expected. That's what they found in the original paper. But one of the things that struck me was that the vast majority of these values are either close to 0 or they're very close to 1. And so, even though there's significant tests, the amount of hybridization is very, very low. Okay, so just to wrap up here, I think inferring hybridization at the population level can certainly be misleading if it's not uniform. And so doing something to sort of account for the fact that there could be heterogeneity in the individual level of introgression is really important. So whether that's testing individuals or doing this kind of bootstrapping that I showed you, hopefully that's something that people will start to do. And the sort of last point generally about phylogenetic invariants is that they're really fast and they're a really powerful way to make inferences about hybridization when you use them sort of more thoughtfully and just very informative for doing things with phylogenetics in general. So I'd like to sort of point out also that all of these inferences that I made were part of the software package that we've been working on that's on GitHub. And then also all of the output files from the analyses and the simulations are part of this tutorial that I've put together for using the software. So that's on GitHub as well. And the slides for this talk will be put on Figshare. So if you want to look at them again, they'll be on. Okay, so also like to thank Cecile and Claudia Solis-Lemus and Ron Fan Cui for help with the swordtail data set. And then, of course, all of my lab mates and NSF and EEOP for funding. So, thank you very much.